Welcome friends, I am Leema Rosemary, Assistant Professor, Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. Now I am going to talk about antigens and antibodies. Antigens means any foreign substance. Here antigens are molecules or molecular structures that are foreign to the body and generally induce an immune reactions in the form of the production of antibodies against them. In simple words, antigens can be anything that does not belong to the body and are foreign. Antigens in humans are proteins, peptides, or polysaccharides. However, lipid and nucleic acid can also access antigens when combined with proteins or polysaccharides. And the properties of antigens, antigenicity, immunogenicity, antigenic determinants, heptans, adjuvants, these are the properties of antigen. Immunogenicity and antigenicity are related but distinct immunological properties. What is immunogenicity? Immunogenicity is the ability to induce a humoral or a cell-mediated immune response. Thus, the substances that induces a specific immune response is called an immunogen, although usually called an antigen. Immunogens are known as complete antigen that elicit a specific immune response by themselves without any carrier particles. These are usually proteins, peptides, or polysaccharides with a high molecular weight component. The molecular weight greater than 10,000 Dalton. The immunogenicity of the antigen increases with the increase in degree of foreignness. Then antigenicity. Antigenicity is the ability to combine specifically with the final product of humoral or cell mediated response, that is antibodies that are secreted. Although all molecules that have the property of immunogenicity also have the property of antigenicity, but the vice versa is not applicable. Then antigenic determinants. It known that immune cells does not recognize or interact with an entire immunogen molecule. Instead, lymphocytes recognizes discrete sites on the molecule that is called epitope or antigenic determinants. Antigenic determinants are regions in an antigen molecule that is involved in the reactions with antibodies. Usually, antigens with two or more antigenic determinants can induce antibody production. Thus, a smaller antigen usually does not induce antibody production as it is not possible for a small molecule to have more than one antigenic determinant. This is the antigenic determinants, this one, this. Heptans are antigens that cannot induce an immune response by themselves. They lack immunogenicity. They are also known as incomplete antigen. These are usually non-protein substances that require a carrier molecule to form a complete antigen. Heptans have low molecular weight and fewer antigenic determinant sites. The carrier molecule bonded to the heptan is considered a non-antigenic component and is a protein or a polysaccharide molecule. Here this is the heptans. It's a small molecule but no immune response. When the heptans combine with carrier molecule to form heptan carrier conjugate, then it induces the immune response. So the chemical coupling of a heptan to a large immunogenic protein called carrier, this yield an immunogenic heptan carrier conjugate. So this is the complete one. So it produce immune response. Animals immunized with such heptan carrier conjugate produces antibodies specific for three types of antigenic determinants. First one for the heptan determinant, then second one for the unaltered epitopes on the carrier molecule, then third for new epitope formed by regions of the both heptan and the carrier molecule. And this is the carrier molecule, this is the heptan, so form complete antigen. So then produced antibodies. Here antibody and heptan carriers, This is the complete one. Then adjuvants. An adjuvant means substances that enhance the immunogenicity of that antigen. It is an ingredient used in some vaccines that helps create a stronger immune response in people receiving the vaccine. They are mixed with an antigens and injected, often used in clinical research to boost the immune response when antigen has low immunogenicity or less amount of antigen available. Then antibodies. Antibodies are protein molecule naturally produced or synthesized by the B lymphocytes. They are also known as immunoglobulin. Antibodies have specific sites that recognize antigen known as antigen binding sites, and that is paratope. These are regions on the antibodies where the antigen binds on the antibody. This is the structure of antibody. Here this is the antigen binding site, that is paratope. This is the epitope. So in the interactions between the epitope and paratope, so produce the immune response. A paratope, also known as antigen binding site, is a part of an antibody which recognizes and binds to an antigen. It's a small region at the tip of the antibody's antigen binding fragment and contains part of the antibody's heavy and light chains. Then antigen antibody interactions. Here the antigen, this is the antigen, this is the antibody. So both interact to form antigen antibody complex. So antigen antibody interactions between the antigens and antibody, these reactions are highly specific, and an antigen reacts only with antibodies produced by itself or with close related antigens. Antibodies recognize molecular shape and antigen. Then the forces of the antigen antibody interactions, that is affinity and avidity, and the combined strength of the non-covalent interactions between single antigen binding site on the antibody and a single epitope is known as the affinity of the antibody for the epitope. Avidity means it's a broader term than affinity. It's a measure of the overall strength of the antigen antibody complex. The strength of multiple antigen antibody interactions between a multivalent antibody and a antigen is called the avidity. This diagram explains affinity and avidity. This is a affinity, so it refers to the strength of a single antibody antigen interactions, but this is the avidity. It refers to the strength of all the interactions. Those are so many antigen and antibody interactions. This is the avidity. Then next, cross reactivity. So all the antigen antibody interactions are highly specific. In some cases, antibody elicited by one antigen can cross react with an unrelated antigen. Such interaction occurs if two different antigens share an identical or very similar epitope. Thus, it's known as cross reactivity. So this is the diagram explain the cross reactivity. Then finally valency. Valency is known as number of binding sites per antibody molecule. All antibodies are multivalent. Example, immunoglobulin G's are bivalent and immunoglobulin M are decavalent. The greater an immunoglobulin's valency, the greater the amount of antigen it can bind. Similarly, antigens can demonstrate multivalency because they can bind to more than one antibody. Multimeric interactions between an antibody and an antigen help their stabilization. Thank you.